Imagine a world where common infections just become untreatable, where, a routine surgery could turn deadly because the simplest bug just laughs at our strongest drugs. Superbugs aren't sci-fi anymore. They're really here, in our hospitals, our neighborhoods, even our bloodstream. And the traditional way we find antibiotics, well, it feels like it's hitting a dead end. So let's dig into this. What if, instead of just searching for medicines that already exist, what if we could actually design entirely new ones from scratch? That's exactly what's happening right now at MIT, actually.
Yeah, and what's really fascinating here is how these generative models, AI drug discovery, are stepping into this critical gap. This isn't just some gimmick. It's a fundamental shift in how we even approach chemistry. So today, we're diving into this groundbreaking study. A team used AI, generative AI, to design two really promising antibiotic candidates. And these are specifically targeted against drug-resistant bacteria, the tough ones. If you look at the bigger picture, this work really points towards a future where we explore that huge chemical space deliberately, by intention, not just by stumbling onto things.
Right. So what does this all mean for you listening? We're going to unpack this problem, the whole antibiotic resistance issue, look at how AI is changing the game, and get a really fascinating inside look at MIT's breakthrough. You'll hear about these two brand new molecules designed by AI, what they target, why it matters. It's quite something for the future of medicine. It's amazing when you think back, antibiotics totally changed human life. Almost overnight, right? Solved infections that were death sentences before, made things routine, but then, yeah, it feels like we hit a wall. Resistance started creeping up. The discovery pipelines just slowed right down. And, well, the economics didn't help either for short-course drugs anyway. Investment dried up. From your perspective, what really led to this stalemate, where most new antibiotics are just kind of tweaks on old ideas?
That's a great question, and it hits the nail on the head. The core issue is that moving in these small, iterative steps, it just doesn't work anymore against bacteria. Why? Because they adapt so much faster than we do. Way faster. We already see catastrophic harm globally from drug-resistant bacteria and traditional discovery. It's too expensive, too slow, which naturally discourages taking big risks. You stick to what you know. So when we do that narrow iterative chemistry, we often land right back in familiar territory. Places bacteria have already figured out how to defend against. It really is a stalemate. We desperately need truly original chemical structures, completely novel ways to attack them, and generative AI drug discovery, it aims straight at both of those needs.
Okay, that idea. Designing molecules from scratch, not just searching. That feels like a huge shift.
Yeah.
How does an AI even start to imagine something completely new, something a human chemist hasn't thought of? What's actually going on there?
It is genuinely revolutionary. So these modern AI models, they learn. They learn from millions, literally millions, of existing chemical structures. They learn bioactivity patterns, what shapes tend to do what in biology, and they learn the fundamental rules of chemistry itself, how atoms bond. Then, using all that learned knowledge, they propose totally new candidates. Molecules that are chemically plausible, they follow the rules, but often they're completely novel structures. And there are, well, two main ways they often do this. Think of fragment-guided generation.
Yeah.
It's sort of like starting with a perfectly shaped Lego brick. As important, maybe it already shows a little activity. And then you get the AI to build the rest of a complex structure around it, making sure all the new pieces fit chemically.
Okay, like building onto a known good starting point.
Exactly. Then there's unconstrained de novo design. This is maybe even more radical. It's like giving the AI all the rules of Legos, how bricks connect, what shapes work, and just saying, okay, build me something brand new from scratch that solves this specific problem. You don't give it a starting piece, just the goal. Both ways are incredibly powerful. They're fast. And crucially, they just blow open the search space way beyond what any human team could ever map out on a whiteboard.
That's a fantastic way to put it. And like you said, this isn't just theory anymore. It's happening. At MIT, these researchers, they didn't just talk about it. They designed, they screened, they actually tested brand new compounds.
Yeah.
And against two really tough bugs, drug-resistant gonorrhea and
Yeah.
MRSA, methicillin-resistant Staph aureus. Their AI platform came up with, tens of millions of possibilities, which they then had to filter down, right, for activity, toxicity, making sure they were genuinely new.
That's precisely it. Tens of millions generated, then filtered computationally. And the best ones, the most promising candidates based on those predictions, were then funneled into actual lab work. Synthesis, making the molecules, and then animal testing. The study reported two key lead candidates that showed in vivo activity, meaning they actually worked in a living organism, not just a test tube. One against gonorrhea, one against MRSA. And this is so much more than just, a bigger database search. It's designing with intent. It builds on MIT's earlier success. You might remember Hallicin, that antibiotic AI found by repurposing a diabetes drug.
Oh, right. Hallison showed AI could find activity in really unexpected places.
Exactly. Hallison showed models could find the needle in an existing haystack. This new work, it goes further. Instead of finding the needle, the team is essentially spinning completely new hay, creating the haystack itself.
Okay, let's zoom in on one of those needles then, NG1, the gonorrhea breakthrough. You said they started with a chemical fragment, something known to have some activity against gonorrhea. Then they used AI tools to build out from that fragment, leading to this candidate, NG1. And what's really exciting here, you mentioned, is its novel mechanism. Can you break that down a bit?
Absolutely. So the experiments are pointing quite strongly towards a protein called LPTA. LPTA is part of the machinery bacteria use to build their outer protective layer, the outer membrane. It's involved in transporting key building blocks, lipoligosaccharides. The evidence, well, they saw things like the LPTA protein becoming unstable when the drug was present, proteomic destabilization. They saw the bacteria changing, which genes were active, which messages were being sent inside the cell in a way that matched the drug dose. And interestingly, NG1 worked even better when paired with another drug, polymyxin B, which also targets the bacterial membrane. That synergy suggests they're hitting related pathways.
Okay, so it messes with the bacteria's outer defenses.
Pretty much. It seems to disrupt that outer membrane integrity or assembly. And crucially, this wasn't just in a dish. In a mouse model simulating vaginal gonorrhea, applying NG1 topically significantly reduced the bacterial numbers compared to the control group. It performed, in the same general ballpark as a standard antibiotic used for comparison. Plus, early safety tests in the lab looked encouraging. No signs of damaging red blood cells or causing DNA mutations at the concentrations they tested. That's a good start.
That is promising.
Yeah.
And then for MRSA with DN1, you said they took a different path, more open-ended.
That's right. For MRSA, they used the other approach we talked about, unconstrained de novo design. They essentially let the generative models design molecules with very few starting structural rules, really let the AI imagine solutions from the ground up. Then again, they computationally filtered that huge list, looking for predicted activity against MRSA, low predicted toxicity, and again, novelty, making sure it wasn't just another known antibiotic structure. From the compounds they actually made and tested, DN1 emerged as a strong candidate. And in a mouse model of MRSA skin infections, specifically in neutropenic mice, which have weakened immune systems, kind of like some vulnerable human patients, DN1 treatment slashed the bacterial burden, like by a factor of 10, an order of magnitude, compared to the untreated control group.
Wow, that's a significant effect in vivo for something designed totally from scratch.
It really is. Getting that kind of signal in an animal model, especially for a de novo design at this early stage, is quite rare and very encouraging. And the mechanistic tests, they showed effects on the bacterial membrane potential, the electrical charge across the membrane, and changes in the cell's shape, all consistent with disrupting the membrane, maybe punching holes in it or destabilizing it. It suggests a different way of killing the bacteria compared to NG1, perhaps a bit broader, less focused on one specific protein target. But the whole process itself, from the AI design through filtering, synthesis, testing, getting that in vivo result, it's not just a scientific finding, it's really a practical roadmap, a pipeline other teams can look at and adapt for building their own AI antibiotics.
Okay, stepping back again, looking at the bigger picture for AI in drug discovery, what does this specific success mean for the whole field? You said it's not a silver bullet, but a force multiplier. What does it multiply? What makes it so powerful?
Well, like you said, it multiplies good questions, good biology, and good chemistry. It takes what we know and scales it up massively. This MIT platform specifically did 3 things that traditional methods really struggle with, especially at scale. First, it just explored enormous chemical spaces. We're talking tens of millions of molecules proposed, many totally new, never seen before in public libraries, real chemical novelty.
Just staggering numbers compared to traditional screening.
Exactly. Second, it optimized specifically for novelty. The filters weren't just looking for activity, they were actively pushing the search away from known chemical structures, known scaffolds. And that's critical, right? Because bacteria have learned to deal with our old tricks. They exploit our chemical habits. We need new angles.
So it forces us out of our old ruts.
Precisely. And third, it allowed them to move quickly to understanding the mechanism. They used techniques like proteomics, studying all the proteins, and transcriptomics, looking at all the RNA messages very early on, plus biophysics. This gave them clues about how the drug was working, like the LPTA connection for NG1 much earlier than usual. It shortens that loop. Design a molecule, test it, figure out how it works, then use that knowledge to design the next molecule better. This is really how AI antibiotics should be built. Design aimed at finding entirely new biology, not just slightly better versions of the same old ideas.
It sounds incredibly powerful, but of course success in the lab, even in mice, is one thing. Getting a drug to people is a whole different journey. What are the big hurdles still ahead for candidates like NG1 and DN1? What's the road look like from here?
Oh, absolutely. There are significant hurdles, no question. But the potential payoff makes tackling them worthwhile. First, there's synthesis and scale-up. Some of these AI-designed molecules can be complex, tricky to make efficiently and affordably in large quantities. That's a real chemistry challenge.
Right, making milligrams in a lab versus kilograms for trials.
Exactly. Then there's pharmacokinetics, or PK, basically what the body does to the drug. How is it absorbed? Where does it go in the body? How quickly is it broken down and removed? Mouse models often use, say, topical application like the NG1 study, or high local doses. For a drug you take as a pill or injection, you need the right PK profile for systemic use in humans. That's a huge step. Safety, of course, is paramount. Those early lab tests are helpful flags, but they're not definitive. You need extensive safety pharmacology and toxicology studies before human trials. Rigorous testing.
The long, expensive part.
The long, expensive, but absolutely essential part. And finally, even if a drug is successful, resistance is always a threat. Bacteria will eventually try to adapt. So the best defense is thinking ahead. Smart stewardship, using the antibiotic wisely, maybe combination therapies, and crucially, starting with something truly novel like these candidates gives you a head start. The good news, though, is that generative AI can give us starting points like NG1 and DN1 that actually justify going through that whole demanding, expensive development process. The potential is that high.
That makes sense. And for the researchers and builders out there, the people actually doing this work, you mentioned this MIT study offers a kind of blueprint. Could you walk us through those key lessons or maybe recommendations for teams wanting to build these AI drug discovery programs?
Sure. I think there are a few clear takeaways. One, start with a really sharp clinical focus. Don't just say, we need antibiotics. Pick a specific pathogen, a specific type of infection where a truly new mechanism could be a game changer. Like the examples here. Gonorrhea with its rising resistance, or MRSA causing serious deep tissue infections. Define what success looks like for the patient, not just in lab numbers.
Be specific about the unmet need.
Exactly. 2, build dual loops, run biology and chemistry in parallel. Use both fragment-guided and unconstrained regeneration. Don't bet on just one approach. And unify them with really strict novelty filters and consistent ways to predict activity. Cast a wide but smart net. 3, bake mechanism investigation in early. Don't wait until you have a lead candidate. Plan for those omics, proteomics, transcriptomics, and synergy tests right from the start. The NG1 story, finding LPTA early, shows how understanding the how can guide the what to design next and how the drug might eventually be used.
So mechanism isn't an afterthought, it's part of the design cycle.
It has to be. 4, design for synthesis. This is crucial. Score your potential candidates not just for activity, but for how easy they might be to actually make. Work closely with medicinal chemists and manufacturing partners early. A brilliant molecule on a computer screen that nobody can synthesize at scale, that's not a drug.
Practicality matters.
Immensely. And finally, number 5, keep clinical reality front and center. Think about the actual use cases. How will this drug be given? Maybe a topical or locally delivered version is a faster path to helping patients, a stepping stone, while you work out the kinks for systemic delivery. The DN1 skin model is a perfect example of that thinking.
That's a really practical set of guidelines. Thanks. Stepping back from the science for a moment, this isn't just about lab results or molecules on a screen. It feels deeply personal. We mentioned gonorrhea. It used to be easy to treat. Now it's often resistant to almost everything. MRSA. It moved out of hospitals into communities. Now it's a major threat in ICUs. These aren't abstract problems. These are the infections that can derail surgeries, make chemotherapy dangerous, turn a simple wound into a crisis.
Absolutely. The human stakes are incredibly high. And that's why this AI drug discovery work is so important. It gives us a credible, powerful way to finally start shifting the momentum back in our favor against these resistant bugs. And the wins here could compound. If we get novel mechanisms, that slows down resistance development overall. If we get purpose-built molecules, maybe they have fewer side effects, less collateral damage. And if better design truly shortens the path to the clinic, you put all that together. It really could look like a second golden age for medicines fighting infection. one that hopefully we can sustain this time around.
Okay, so let's quickly recap what we've unpacked today. AI drug discovery is making this fundamental shift, moving from just searching existing molecules to actively designing completely new ones. This massively expands the chemical possibilities and really unlocks novelty. We saw how MIT's platform put this into action, producing two promising leads that work in vivo. NG1 for gonorrhea, which seems to target the LPTA protein,
Right, involved in building that outer membrane.
and DN1 for MRSA, which looks like it disrupts the membrane more generally. We also learned how vital it is to figure out the mechanism early. It de-risks development and helps figure out how the drug might actually be used. This builds on earlier AI wins like Allison, and it really points towards a more resilient future for antibiotics powered by AI.
Exactly. And it's important to remember, AI drug discovery isn't magic, it's a method, a powerful one. It lets us ask better questions of biology, explore vastly more chemistry, and move faster with real purpose. The sooner we really embrace building with these tools, the sooner we can hopefully deliver therapies that actually work against these growing threats to patients.
Yeah, this work from MIT definitely shows the path is real. It's not just theoretical anymore, it's open for development. So, final thought for you to mull over. Thinking about this new era of AI-driven discovery, what kind of tough pathogen or challenging data set or specific clinical situation where resistance is currently winning? What would you bring forward to tackle with these powerful new tools? Something to think about until our next deep dive.